Before I share my screen, let me state that I've done some grading and I know I've got some labs graded. Let me see if I can go there. Close that. I got the last lecture worksheet graded too. I don't remember the name of that one though, but oh, and I got 2 extra credit study graded. Extra credit study 2 and extra credit study 3. I got worksheet lecture 3 graded. And I have lab 00, lab 01, lab 2, and lab 3 graded both the first and the second time. For lab 4, I've only graded it the first time, but actually I think you still have time to make corrections. Yeah, you can make corrections till April 29th on lab 4. Any question about any of the grading? Oops, sorry, I lost my mouse there. All right, no questions. Let me get out of the grade book and then I'll share my screen. I think I got the extra credit quiz graded, and let me state that if you're not seeing the score and extra credit quiz 1 or 0, whatever that one is, just click on the icon. The score is there, and for some reason it's not showing. I've ran into this before and I don't know what the problem is because the grade is there. It is graded and you got to click on the icon to see the grade. It does not count in the current points, but it counts in the total points. So you have to look at the right, what do you call it, column to see the points counting. All right, I think that's it, but let me look to see if there's anything I should mention. Share my screen now. So today is April 27th, and we should be starting chapter 4. I'm pretty sure we did finish chapter 3, but let me look on that. Do note that you have a quiz this Wednesday, quiz 2. It'll cover labs 3 and 4, chapter 3, and the end of chapter 2. And that reminds me now what I wanted to talk to you about in the grade book. I put in a discussion board under week 4 lecture quiz discussion. Please click on it and you'll get a discussion board for discussing when do you want to have the quizzes. We currently have them 8 p.m. Tuesday night to 8 p.m. Wednesday. Some students have asked for it to be switched to 11:59 p.m. Tuesday to 11:59 p.m. Wednesday. Okay. Other times are possible, just bring it up in the discussion. And then after people have discussed, vote on it. You can access this. That doesn't look right. I thought I changed the date there. To May 3rd. Okay, that's right. So why is that not showing that in the grade book? Anyways, you can access that. I think that's midnight tonight till May 3rd. Yeah, see, that's not showing right. Oh, well. Um, oh, and state if there's a time that does not work for you and you must state why it doesn't work for you, or else I will not consider uh, your comment of why a certain time will not work for you. All right, I think that's it. Check to make sure. We do have a lab today, lab 5, streaking for isolation and culture media. That's not right. We've already done that lab. Today is Thursday, so we're doing lab 6, disc diffusion and antibiotics. And then read in the lab, read the unknown project instructions. I'll discuss the unknown project next week. And that'll be Tuesday. We'll have lab 7 on Thursday. All right, any questions about what we're doing? Hopefully we'll finish chapter 4 today. I think I got that up already. All right, I have in my notes that we had talked about the endoflagella. Is that right? All right, I'm not hearing anything, so I'm just going to start from here and then start talking about other structures in prokaryotic cells. The fimbriae are hair-like structures that some bacteria have. They are shorter and straighter than flagella. They are not used for swimming, so not really used for locomotion. They are used for attaching to surfaces. Any question about any of that? The pili are also called the sex pili, are longer than the fimbriae. And usually the cell only has one. I've never seen one that had two. In fact, if you look closely here, let me see if I can blow this up. You can see that the cell that has the sex pili right here does have fimbriae. Now you can see it. So the fimbriae are quite shorter than the sex pili. The sex pili is a sort of a reproductive structure, and it's used to transfer DNA from this cell, which has the sex pili, into that cell, which does not have the sex pili. And pili is plural, pilus is singular. Any question about any of that? We'll talk about the sex pilus in chapter 8. The prokaryotic cell wall is another cell structure we need to talk about. It is a complex, semi-rigid structure, and it's responsible for giving bacteria their cell shape. The cell wall protects the cell and prevents osmotic lysis. For example, a red blood cell, and actually any animal cell, lacks a cell wall. If you were to put red blood cells in a beaker of water, the water will move into the red blood cell. The cell membrane would expand, and it would just keep expanding until the membrane burst, and then the cytoplasm would leak out of the cell, and that would kill the cell. For a prokaryotic cell, and actually any plant that has a functional cell wall, if you put those cells in water, water will move into the cell, but only to the point where the cell membrane expands up to the cell wall. Then the cell membrane cannot expand any further, and that will prevent water from moving into the cell because there's no room for it to move into the cell. And that prevents osmotic lysis. It's extremely important in bacteria for protecting their cells. Any question about any of that? We'll actually talk a little bit about antibiotics and how penicillin G, it actually creates defects in the cell wall, which allow the cell membrane to then protrude out of that defect. And then water can move into the cell and the cell membrane protrudes further until it expands, and that'll kill the cell. The cell wall is located outside of the cell membrane, and it is considered a part of the cell. Anything beyond the cell wall is not considered a part of the cell. And it's kind of weird that the cell wall is considered a part of the cell because it is external to the cell membrane, and the cell membrane is the gatekeeper deciding what can enter or leave the cell. So it's the one determining what can enter and leave the cell. But the cell wall is considered a part of the cell, and it is external to the cell membrane. The cell wall does contribute to the pathogenicity of some species, like Mycobacterium, like Mycobacterium tuberculosis. The cell wall does contribute to this pathogenicity of the species. The cell wall is mainly made up of peptidoglycan, at least in bacteria. Now, it is true that the archaebacteria do have a cell wall. However, their cell wall is not made up of peptidoglycan. Any questions about any of that? All right. Peptidoglycan is the main molecule in the cell wall of bacteria. It's a polymer of a repeating disaccharide, and you can just abbreviate the disaccharides, meaning the two sugars, as NAG and NAM. Sorry. NAG stands for N-acetylglucosamine. NAM stands for N-acetylmuramic acid. And those names probably mean as much to you as NAG and NAM. You can call them by either name. The disaccharide is linked together, meaning the disaccharide goes one after the other. Let me see if I can blow this up. It's a chain of NAG and NAM, and maybe this would be the best one to show it on. Come on, mouse. There's NAM, followed by NAG, followed by NAM, followed by NAG, followed by NAM, etc. And that makes a row. And then there's rows next to it on a plane. And then underneath, there's another plane and another plane. And usually there's a plane above it too. Okay. And the disaccharide is hooked together by short polypeptides. This polypeptide here, come on, mouse, connects this row of NAG and NAM with this row. And then we have a short polypeptide here connecting that polypeptide to that polypeptide. And that links them together in the cross-link. So this one is linked to that one by this part of the polypeptide, that polypeptide, and that polypeptide. This gives peptidoglycan its name. Glycan means sugars, and there's two sugars in peptidoglycan, the NAG and the NAM, and then pepto from the short peptides, peptidoglycan. Penicillin actually inhibits this lattice formation of the peptidoglycan. It actually gets in between the cell wall when it's growing, and laying down the cell wall. And before the peptides get put in, right here and right here, the penicillin gets here and then it blocks this peptide from forming. And that will create a hole in the cell wall. And the cell membrane can protrude out of this hole right here. Any question about any of that? And that's how penicillin G, as well as all members of the penicillin family, kill cells. They prevent the peptides from linking when the cell wall is being formed. And then there will be a gap in the cell wall right here where the cell membrane can protrude out of this gap. And then water can move into the cell, and it'll just keep moving into the cell because the cell membrane will expand out of this hole. Come on, mouse. Keep losing my mouse. So the cell membrane will expand out of this hole. It'll just keep expanding until the cell membrane bursts. Any question about any of that? All right. The gram-positive cell wall does differ from the gram-negative cell wall. So let's look at that once more. The purple is the cell wall of gram-positive cell. And you can see it's pretty thick. Below that's the yellow of the cell membrane, and it's much thinner. And there we see several layers of peptidoglycan. In reality, it's many more than 3 layers. Let's say it's on the neighborhood of at least 1,000 layers of peptidoglycan, 1 layer on top of each other, for most gram-positive cells. You'll note that there are some other molecules besides peptidoglycan, and obviously the cell membrane. In the cell wall, there's more than just peptidoglycan. In the gram-positive cells, we have teichoic acid coming here, and then teichoic acid hooked to a lipid, and lipoteichoic acid does run through the cell wall, and then the lipid portion runs through the cell membrane. It's thought that the teichoic acid helps ions move through the cell wall. Now, an ion can dissolve in water, and anything that can dissolve in water can go through the cell wall. But the ions can move down the teichoic acid. So it's thought that the teichoic acid, as well as the lipoteichoic acid, helps the ions move through the cell wall. Any question about any of that? The teichoic acid is thought to, as well as the lipoteichoic acid, is thought to help hold the peptidoglycan layers in place. So it sort of helps anchor the peptidoglycan. Yeah. Polysaccharides, meaning the teichoic acid, provides antigenic variation. So some cells will have different antigens depending on the makeup of their teichoic acid. Any questions about any of that? All right, the gram-negative bacterial cell wall differs, and it does have the cell membrane, obviously. It does have a polysaccharide, excuse me, not a polysaccharide, drawing a blank now. The peptidoglycan, it does have a peptidoglycan layer, but it's much thinner than the peptidoglycan layer in the gram-positive cells. And you can note here that it is thinner, but it's more than 1 layer thick. Okay, this slide is only showing 1 layer, but it is more than 1 layer. It's several layers up until someplace around 20 layers of peptidoglycan in the gram-negative bacterial cell wall. And there is more than just peptidoglycan in that cell wall. It does have an outer lipid layer shown here. And you'll note that the main molecule in this lipid layer of the cell wall, which we call the outer membrane of the cell wall, because this is a membrane, that the main molecule in the outer membrane is not peptidoglycan. Not doing too well today. It's phospholipid. It's the same molecule as making the cell membrane. That's the main molecule in the outer membrane. In fact, this layer of the outer membrane of the cell wall is mainly phospholipid. This layer, though, does have phospholipid in it, but it does have another lipid in it, the blue one with that blue hair. The lipid A is this portion here, and then this is an O polysaccharide tail linked to the lipid A, so this is called the lipopolysaccharide. Any questions about that? Now, lipid A is important because it is an endotoxin. And toxins, if you do not know, are things that are harmful to us. So you don't want to consume a toxin. But every time you do consume a gram-negative cell, you will get some lipid A. And then every time you have a gram-negative infection, you are exposed to lipid A. Any question about any of that? And lipid A is an endotoxin, so we don't really want exposure to it. All right. There are other molecules in the outer membrane. The most important is the porin protein, shown here. The porin protein is important to the gram-negative cells because molecules have to move through the outer membrane as well as the cell membrane if the cell wants those molecules. And then molecules that want to leave the cell have to move through the cell membrane and the outer membrane of the cell wall. For the movement of molecules through the outer membrane, they get through the outer membrane by going through the porin protein. So the porin protein is a channel, and many molecules can move through it. Not all, but many. That differs for the molecules moving across the cell membrane. If a molecule wants to move across the cell membrane, it needs an active transport protein to move it across the cell membrane. So any molecule wanting to come in, like glucose or amino acid, or I don't know, amino acid or glucose, or let's say a nucleotide, it will come through the porin protein for getting through the outer membrane. Come on, mouse. But for getting through the cell membrane, it has to find an active transport protein that can move that one molecule. And active transport proteins generally only move one molecule. So we'd have a different active transport protein to move glucose, or to move lactose, or to move fructose. We'd have a different active transport protein to move it across the cell membrane. But all of those will move through the porin protein, as well as amino acids and nucleotides. They'll all move through the porin protein. So the porin protein is very different than an active transport protein, because an active transport protein, generally speaking, only moves one molecule, and only that one molecule. The porin protein moves large numbers of molecules. Any question about any of that? You'll note that, oh, I've got this in bigger picture here. There are lipoproteins in the cell wall of the gram-negative cell, and the lipid portion will be in the outer membrane, and perhaps in the cell membrane if the lipoprotein combines with both. And then the non-lipid protein will be in the peptidoglycan layer. So on the gram-negative cell wall, we have an outer membrane. It does contain phospholipids. That's the main molecule. It does have a lipopolysaccharide, and that will include lipid A as part of that. And lipid A is the endotoxin, the most important part. And then it can have lipoproteins. The gram-negative outer membrane can provide protection to the cell. Certain antibiotics cannot get through the outer membrane. Certain complement proteins cannot get through the outer membrane of the gram-negative cell as well. The antibiotic we're going to talk about is penicillin G. Cannot get through the outer membrane of the gram-negative cell, and penicillin G interferes with the peptidoglycan layer when it's being formed, and so penicillin G does not harm gram-negative cells. It doesn't work on them. Complement proteins generally do not work on gram-negative cells as well. The O polysaccharide is an antigen, which is important to, that's the hair on the lipid A, the O polysaccharide, the part of the lipopolysaccharide, which gives the cell its full name. Like when we're talking about E. coli, O157H7, H7 and O157 are antigens, and O157 is the specific makeup of this O polysaccharide on E. coli. If you don't know, E. coli O157H7 is a pathogenic E. coli. People who get this strain of E. coli tend to have gastroenteritis. It's not a normal strain of E. coli, which lacks O157, and the normal one also lacks H7. The normal one, they don't give its name. They just call it E. coli, meaning they don't give antigens to it. All right, any question about any of that? If not, let's look at the comparison of the gram-positive cell walls and the gram-negative cell walls. Gram-positive cell walls have thick peptidoglycan. It gives it a rigid structure. The gram-negative cell wall has a thinner peptidoglycan layer, and it's less rigid. Teichoic acid is found in the gram-positive cells. There's no teichoic acid in the gram-negative cells. There's no outer membrane in the gram-positive cells, and there is an outer membrane in the gram-negative cells. And that outer membrane does have lipid A in it, which is an endotoxin. Any question about any of that? All right. The difference in the the differences in the cell walls, the gram-positive and gram-negative, give rise to the gram stain. The crystal violet iodine complex forms and does not wash out of the thick peptidoglycan layer of the gram-positive cells. It does wash out of the thinner peptidoglycan layer of the gram-negative cells. And then the alcohol of the decolorizing step dissolves the outer membrane, and that way the safranin can get into the cell and then stain the cells pink. Any question about any of that? All right, this slide showing you a little bit of a summary of the gram-positive cells. Gram-positive and gram-negative. Gram-positive retain crystal violet iodine complex. The gram-negatives do not retain the crystal violet iodine complex, and so they will stain pink with the counter stain. The gram-positive stain purple with crystal violet. They have a thick peptidoglycan layer, and they have a thin peptidoglycan layer for the gram-negative. This says a single layered. This table is not correct. It is several layers. Okay, it may only be 3, but it generally is up to about 20 layers of peptidoglycan. It's thin, much thinner than this one. Teichoic acid is found in the gram-positive, absent in the gram-negative. You don't need to know about the periplasmic space. The outer membrane is absent in the gram-positive, present in the gram-negative. And then the lipopolysaccharide containing lipid A is absent in the gram-positive and is present in the gram-negative. There are some cells that have atypical cell walls, and we've actually already talked about some of them. Let me start with the second one first. The archaea are different from the bacteria. They're a different domain. When we're talking about the bacterial cell wall, the archaea do not have that cell wall. The archaea do tend to have cell walls. They're made of pseudomurine, which is another disaccharide, but it is not peptidoglycan. So the archaea have a different cell wall than the bacteria. And some of the archaea lack cell walls. I could say that there is one family of bacteria that lack a cell wall as well. And we'll talk briefly about that family later in the term. It's the mycoplasma. The mycoplasma family lacks cell walls, and they're bacteria. We've also mentioned briefly the mycoplasmas. Excuse me, not the mycoplasma, the mycobacterium. These are considered gram-positive cells, but they're different than normal gram-positive cells. Let me see if I can go to Google and show you that. If I don't get it quickly, I won't show you. Yep, why are we so big? Well, this will be good enough. A little complex, but oops, well, I don't know if I go one more if it'll show. Go back. Oh, there it is. All right. It's not labeled here, but that's it there. This is the cell membrane, and this is the cell wall. We have a different makeup of the cell wall. There is a layer of peptidoglycan, and I'm having a hard time reading this because I'm not sure what LAM stands for, and I can't even read that there. Oh, here it is. Let's go to that one. There's the mycobacterium cell membrane. There's the peptidoglycan layer, and it does have a fairly thick peptidoglycan layer, not as thick as most other gram-positive cells, but it is a gram-positive cell. And then it has this right here, an external molecule to the peptidoglycan, and this is mycolic acid, and there's a lot of mycolic acid in the cell wall of the mycobacterium. And that gives these cells different properties from gram-positive cells. they have acid-fast staining because of all that mycolic acid in the cell wall, meaning the acid-fast stain can come into these cells, and when you decolorize, it doesn't remove it from the mycobacterium, and it does remove it from other bacteria. Okay, so these cells have a different cell wall because of the large amount of mycolic acid in the cell wall. Mycolic acid is a lipid, and it prevents molecules which are dissolved in water from actually getting in the mycobacterium. And that could be the gram stain, for example. And then the other bacteria with an atypical cell wall are the mycoplasmas. I did mention it briefly just a little while ago, and they don't have a cell wall. The mycoplasmas are the one family of bacteria that lack a cell wall. Curiously, they have sterols in their plasma membrane to give support and strength to the plasma membrane. And it's true that animal cells, which have only a cell membrane, have sterols in their plasma membrane to give it strength and support. Our sterol in animal cells is cholesterol. In the mycoplasmas, it's another sterol. Any question about any of that? All right, you can damage the prokaryotic cell walls with lysozyme, which actually degrades the disaccharide and peptidoglycan, or you can damage it with penicillin. I've already talked about that. It inhibits the peptide bridges in the peptidoglycan, creating holes in the cell wall. You don't need to know the names of the different cells, but you should know that lysozyme is an enzyme that does degrade the bacteria cell wall. And actually, lysozyme is an enzyme made by our skin cells and our not eye cells, but in our tears and some of our mucous membranes. And we secrete this enzyme to help protect us from bacteria. All right. Damage of prokaryotic cell walls, the gram-negative bacteria are not as susceptible to damage of the cell wall compared to gram-positives. This is especially true when we're talking about penicillin G, because that'll damage the cell wall of a gram-positive cell, but it does not work on gram-negative cells. There are some other antibiotics that can work on gram-negative cells, such as streptomycin, that can penetrate the outer membrane. Streptomycin is a lipid-soluble antibiotic, so it can get right through the outer membrane of the gram-negative cell wall. And then there's other members of the penicillin family or other antibiotics which can work on the gram-negative cells. So don't tell me the gram-negative cells are resistant to penicillin, because it's only true for penicillin G and penicillin V and a few other members of the penicillin family. Ampicillin, which I think was the third or one of the early members of the penicillin family to be made, actually works on gram-negative bacteria. Ampicillin and carboxycillin and many of the recent members of the penicillin family work on both gram-negatives and gram-positives. Any question about any of that? All right.
I had a question about the slime layer that's in chapter 4. Which ones are more likely to form slime layers, gram positives or gram negatives?
That's a good question. I'll have to look it up because I don't know that off the top of my head. Okay, so let me write that down and I'll try and look that up. Another thing is that slime layers is not something I work with because you can't see them under a light microscope. And I've never worked with slime layers or an electron microscope, which you need to see the slime layer. Now you can have a slime layer on both a gram positive and a gram negative. That I know, but I'm not sure which one is more likely to have a slime layer. All right, let's move on to the plasma membrane. The plasma membrane is mainly made up of the phospholipid molecule, and that phospholipid molecule gets together to form a bilayer like this. Let me see if I can blow this up. Or like that. There are other molecules in the plasma membrane, such as proteins, and there are 3 different types of proteins. You can have other molecules in here like carbohydrates too, which are not shown. We're only looking at the 3 different types of proteins. You can have peripheral proteins, and they are a protein that are mainly on one side of the membrane or the other side. And that's why it's called the peripheral protein. You can have integral proteins and then transmembrane proteins. Both of these proteins span the entire length of the membrane. We've talked about channel proteins. That looks sort of like a channel protein. And we've talked about active transport proteins. They span the membrane. The plasma membrane is actually a fluid, and we use the fluid mosaic model to discuss and theorize about the cell membrane. So the membrane is a mosaic of phospholipids and proteins, and the membrane is as fluid as olive oil. We're talking about olive oil at room temperature. Obviously, if you put olive oil in the refrigerator, it will become either partially solid or completely solid. And it's kind of hard to think about it because I'm an animal. All I have are cells with cell membranes holding the cells together. And if you look at my hand, it's being held together by a fluid, the cell membrane. Proteins can move in this fluid, and some proteins move to function. Those of you who have taken anatomy and physiology, you know that when a ligand binds to a receptor protein, that those receptor proteins tend to move together before they come into the cell. And the point is that many of the proteins can actually move about in the cell membrane. The only proteins that do not move about are the proteins that are anchored either outside the cell to an extracellular matrix. Like if this was a bone cell, we have the extracellular matrix of the bone out here, and that protein can be anchored to the extracellular matrix. This protein will then not move. Another way we can have it is if the protein is anchored, let's say this one here, is anchored to the extracellular matrix, the intracellular matrix, and then this protein won't move about. This protein, which is not anchored, obviously can move in this fluid. The phospholipids also rotate around and then move laterally. It's actually important for the movement of water into a cell, assuming that the cell wants water, meaning there's more water outside, so it tends to move inside. Water cannot move through the lipid bilayer of the cell membrane. However, water is small enough that when this phospholipid moves here, water can sneak through and then come down to here before this molecule moves over into that position. And the water can't get through this layer of the bilayer, but the water just waits right here until this phospholipid moves over, and then that water can move through. Okay. Now, obviously, that would be too slow for the movement of water into a plant for something like its roots. And so we have active transport proteins in the roots of, well, at least most plants, if not all plants, to move water from outside the cell into the cell. And maybe I shouldn't say an active transport, maybe I should say a channel protein, because the water moves into the cell without requiring energy, but it moves through the channel protein. Any questions about any of that?
And are there aquaporins in the bacteria as well, like in the plants?
You know, I know aquaporins are in plants. I'm not sure if aquaporins are in bacteria. So let me look that one up too.
Okay, thanks.
For those of you who are taking these questions, obviously you don't need to know these answers, and we're not even going to test you on what an aquaporin is, but that's actually the channel protein of plants that I had mentioned. All right. The plasma membrane shows selective permeability. This means it allows the passage of some molecules, but not other molecules, to move across the cell membrane. Large molecules, especially large protein, carbohydrate, or nucleic acid molecules, cannot move across the plasma membrane without the help of a transport protein. Ions either do not move across the plasma membrane or they move very, very slowly. And that's because an ion, even though it's very small, it can't move through the lipid bilayer here. Lipid molecules can move right through the lipid bilayer. So a lipid molecule can move into or out of a cell. However, if you think about it like a cell in the blood, there's not going to be a lot of lipids in the blood because the blood is mostly water. There are some lipids, as you know, cholesterol, and what's the other things? Lipoproteins, what's some of those things you find in the blood? Lipoproteins, I don't remember. There are some. And anyways, the point is that lipids can move right through the cell membrane. Smaller molecules, especially small nonpolar molecules, can move right through the cell membrane, and they actually diffuse across it. For these small molecules we're talking about are oxygen and carbon dioxide, and I think you know that oxygen has to get into the cell from outside, and it just moves right through the plasma membrane by diffusion, and then carbon dioxide must move out of the cell, and it does. It just moves through it by diffusion. And I already talked about water. Can move in, but not easily. The plasma membrane does have a number of specialized functions, especially in bacteria. For example, in bacteria, we find the enzymes for making ATP in the bacteria. In eukaryotes, we do not find this in the cell membrane. We actually find this, the enzymes for making ATP in the mitochondria. And we'll talk more about that later. For the photosynthetic bacteria, come on, we have the photosynthetic pigments found in the cell membrane. They're actually found in infolding of the cell membrane. So this cell membrane comes here and then circles around like that, then comes here, circles around like this, circles around like that. And in these infoldings of the cell membrane, we have the photosynthetic pigments. You can call them chromatophores or thylakoids. It's similar to the terms we have in eukaryotes. We do not find the photosynthetic pigments in the cell membrane of eukaryotes. They are in the chloroplast of eukaryotes. And then, of course, damage to the plasma membrane will cause leakage of the cytoplasm, and that can occur with either alcohols, quaternary ammonium compounds, or polymyxin antibiotics like polymyxin B puts a hole in the cell membrane, and then the cytoplasm can leak out of that hole. And if the cytoplasm leaks out of the cell, then the cell will die. Any questions about any of that? All right, the movement of materials is extremely important to all cells. The movement of material across the plasma membrane can happen by two processes. It can be a passive process or an active process. The passive process means the molecule moves from an area where it's concentrated to an area where it's less concentrated, or we say the molecule moves down its concentration gradient. And there's three ways we can have a passive movement of material across the cell membrane. You can have simple diffusion, where a solute moves across the cell membrane, and in the solute would be something like oxygen or carbon dioxide, and the solvent that the solute is dissolved in would simply be water. You can have facilitated diffusion, which is diffusion. However, the diffusion is occurring through a channel protein, so we call it facilitated diffusion, where the molecule moves down its concentration gradient, but it can only move through this channel protein into the cell. And then lastly, there's osmosis. Osmosis is the movement of water. Water is not a solute, it is a solvent. And osmosis is very important to living systems. So we have a term for osmosis, which is osmosis. And that's the movement of water from areas of higher concentration to areas of lower concentration. And it is the movement of water across a membrane. Okay, any questions about the passive movement of a molecule? The active movement is where the molecule moves against its concentration gradient. So we have sugar at low concentration outside the cell and sugar at high concentration inside the cell. The sugar outside the cell can move by active transport into the cell, and this requires a transport protein to move the sugar into the cell, and this costs the cell one ATP molecule to move one molecule against its concentration gradient. Any question about any of that? If not, let's talk about these in a little more detail. Diffusion is the movement of a solid from an area of high concentration to an area of low concentration, such as when we put the dye of ink in the water. Concentrated ink will move by diffusion to other areas, and given enough time, this dark purple will be gone, and the water will all be uniformly light purple. Oxygen and carbon dioxide move by simple diffusion across the cell membrane of cells. Facilitated diffusion uses a channel protein, and that's called a transporter protein here. And the substance only moves from areas of high concentration to areas of low concentration. This chart is showing you glucose, but I want to switch it to fructose because glucose works a little differently than this chart is showing. But fructose, when you eat fruit, for example, in your gut, you'll have a high concentration of fructose, and inside the cell, there will be a low concentration of fructose. So fructose will move through this channel protein by facilitated diffusion from areas of high concentration to areas of low concentration. What this means is that not all of the fructose will ever be digested, because once the fructose outside the cell equals inside the cell, no more fructose will move into the cell. The advantage of this is that this costs the cell no energy to move fructose into the cell. Any question about any of that? All right. In both simple and facilitated diffusion, we're talking about solutes moving across the membrane, and they move down their concentration gradient from areas of high concentration to areas of low concentration. Osmosis is the net movement of water, which is a solvent, not a solute. The water molecules move from areas of high concentration to areas of low concentration. You should realize that when we're discussing tonicity, it is not defined by water. It is defined by the concentration of the solute. Wherever we have high concentrations of water, we have low concentrations of solute, and vice versa. Whenever we have low concentrations of solvent, we have high concentrations of solute. And we'll talk more about the tonicity in a future slide. Actually, it's this one. You can have a cell in 3 tonicities: isotonic, hypertonic, and hypotonic. I was looking at the time here and saying it's almost finished, but then I remembered this clock is wrong. We have about 20 more minutes. You should note that the tonicity is a relative term. So when we're saying the solution outside the cell is hypertonic relative to the solution inside the cell. Any questions about any of that? All right, isotonic is the easiest to discuss, and that's where the concentration of solute is equal outside the cell compared to inside the cell. Isotonic is, in all the tonicities, is defined by the concentration of the solute. Wherever the solute is high, then the solvent is low. In this case, the concentration of water will be equal outside the cell to inside the cell. So there will be no net movement of water into or out of the cell. Notice how I worded that. I said no net movement of water. That's not saying that there's no movement of water. It's just for every water molecule moving into the cell, there would be one moving out of the cell. No net movement of water. The cells are happy in their isotonic solution, that means they can survive. And as you know, your blood cells are in an isotonic solution because otherwise your red blood cells would lyse if they weren't in an isotonic solution. Either water would enter the cell or water would leave the cell. A hypotonic solution is when the solute concentration outside the cell is lower than inside the cell. This means that water will move into the cell. Note that we're defining the tonicity by the solute, but it's actually the water that moves, not necessarily the solute. So in a hypotonic solution, water will move into the cell. If the cell does not have a cell wall, water will move into the cell, expanding the cell membrane until the cell membrane will pop. And of course, that will kill the cell. Most bacteria actually live in such a solution, a hypotonic solution. And that's why bacteria, like when they invade the body, they need a cell wall to protect them from a hypotonic solution that we call osmotic lysis. All right, any questions about that? All right, a hypertonic solution is where the concentration of solute outside the cell is higher than inside the cell. This will result in the net movement of water out of the cell. That means the cell membrane will pull away from the cell wall, will shrink, and this can lead to plasmolysis. Generally, this causes cell death, like E. coli will not survive very long in a hypertonic solution. It depends on what the cell is, like a endospore can live for decades, maybe even millennia in a hypertonic solution. But most bacteria, this would lead to cell death, something like E. coli, and it will die before a day. I'm not sure how long it would take before E. coli would die, but it would die fairly quickly. Less than a day, maybe even less than an hour, but I'm not sure. And that is why when you have molasses, you can keep it at room temperature for eons. I'll tell you a story that my mother had a bottle of molasses for 30 years before she finally decided, I should say at least 30 years, before she decided to eventually throw it away. And what happened is when the bacteria fell into the molasses have such high concentration outside the cell that the water would leave the cell, and that would prevent the bacteria from growing and would tend to kill the bacteria. All right. So passive processes are okay when the concentration of the substance required by the cell is higher outside the cell than inside the cell. Like when your cell wants fructose and you just ate a high fruit diet, then the passive processors are fine. However, what do you do when the concentration of the molecules you want outside the cell are very low and much lower than the concentration of those molecules inside the cell, such as a cell living in the open ocean or in the desert? It's not going to find glucose or fructose very often in the water or in the desert sands. So the cell resorts to active transport to move the molecule of interest into the cell against its concentration gradient. This requires the use of energy by the cell to move the molecule against its concentration gradient. So the cell needs to use work to move the molecule into the cell. And the cell can do this by using an active transport protein, which actually pumps the molecule against its concentration gradient. This requires energy so that active transport protein will move one molecule, and only that one type of molecule, like glucose, into the cell, but it costs the cell energy, and it takes one ATP molecule to move in one molecule against its concentration gradient. Any question about any of that? All right. So bacteria have another way of moving molecules into the cell that is similar to active transport, but it's a little different. And we call this mechanism of moving molecules into the cell against their concentration gradient, we call it group translocation. It's a special form of active transport. It is only found in prokaryotic cells. It does require a transport protein. It does require a high energy phosphate compound, which is usually not ATP. An example would be PEP, where the phosphate comes off of the PEP and then binds to the molecule that the cell is moving into the cell. This movement of the molecule into the cell chemically alters the molecule while it is moving into the cell. For example, when the cell wants to move glucose into the cell by group translocation, the phosphate will be used up, giving the cell the energy it needs to transport the glucose against its concentration gradient, meaning PEP, when it separates and gives off the phosphate, that releases energy. And then that phosphate group coming off of PEP will actually bind to the glucose, chemically altering glucose from glucose to glucose 6-phosphate. This alteration helps prevent escape of the molecule from the cell and allows the cell to more easily accumulate substances. For example, let's say we had 10 molecules of glucose already inside the cell, and we'll say 9 molecules of glucose outside the cell. So you need to move a glucose in by either active transport or group translocation, because the glucose is going to move against its concentration gradient. Active transport, I think you understand, the cell will move the glucose into the cell, and that'll cost the cell 1 ATP, and then we'll have glucose into the cell. Group translocation, though, if we move, let's say, 5 glucose molecules into the cell by active transport, and we had 10 to begin with, we now have 15 glucose molecules in the cell. And if we move the other 4 by group translocation into the cell, let's talk about the 4th one. You're moving it against its concentration gradient, but it would be easier to move it against the concentration gradient where there's 1 outside the cell and 4 inside the cell. It's easier to move it into the cell against 1 into 4 than it would be to move it by active transport, because there's only 1 outside the cell and 15 inside the cell. And it's thought that prokaryotes have group translocation to help them move molecules into the cell more easily than just using active transport alone. Any question about any of that? All right, the important thing is group translocation moves molecules against its concentration gradient. It does cost the cell energy. And then the molecule moved into the cell is chemically altered. Check the time here. Oh, still have time. The cytoplasm is the substance inside the cell. It is mostly water. About 80% of the cytoplasm is water, and then the remaining are dissolved molecules that we call solutes in the water. The ribosomes, the inclusions, are molecules that are so large that they are not dissolved in the cytoplasm. The nucleoid region contains the DNA, but wherever the DNA molecule floats to in the cytoplasm is a region that we call the nucleoid. There is no boundary between the nucleoid region and the cytoplasm because there's no membrane to separate them. Our ribosomes and inclusions are found in the cytoplasm, but they're too big to be dissolved in the cytoplasm. All other molecules, at least all other molecules that I know of, are dissolved in the cytoplasm. And that would include most of the time DNA, proteins, nucleic acids, amino acids, glucose, all those other molecules. All right, I've already talked about the nucleoid region. Let me state that we can have some DNA, which is outside the chromosome of a bacteria. And just for your information, you can have this DNA outside the chromosome in yeast. And we call this DNA, a plasmid. A plasmid is just a short piece of DNA that's not part of the chromosome. The chromosome is a piece of DNA, but it's a very long piece of DNA. Ribosomes are the site of protein synthesis in cells, and they're made up of RNA, ribosomal RNA, and then ribosomal proteins. They are so large that they do not go into solution. So they are not in solution of the cytoplasm. And you can actually see them here, and they're the dots in this picture here, which is an actual electron microscopic image. You don't need to know that ribosomes have different units or know the names of these units, but you should know that ribosomes are the site of protein synthesis and that the prokaryotic ribosome is a little smaller than the eukaryotic ribosome. The prokaryotic ribosome is 70S. The S stands for sedimentation coefficient, and this is where the ribosome sediments in the ultracentrifuge gradient, the 70S location. And it's just a way that biochemists name or configure ribosomes and other things. Any questions about any of that? The eukaryote ribosome is a little bigger, and its size is 80S. Inclusions are reserve deposits within the cytoplasm that accumulate to such levels that they precipitate out of the cytoplasm. The cell tends to use them when resources are low. So inclusions oftentimes include products that the cell can metabolize. You don't need to know the different names of inclusions, but you should know that there are different types of inclusions. Magnetosomes is an inclusion here. And like I said, an inclusion is just something the cell is accumulating, and it accumulates it at a level so high that the molecules precipitate out of solution of the cytoplasm. And then we see the inclusion under the light microscope. This bacteria obviously metabolizes sulfur, so it brings sulfur into the cell and then just stores it. And so much sulfur is stored that inclusions form. And there's a metachromatic granule of an inclusion. Let me see if I can blow that up. There you can see one cell here and has 3 different inclusions. Endospores are a special structure that some cells make to help them survive. So the endospore is a survival strategy. It is not a reproductive strategy. The endospore is a specialized resting cell that is produced by a few species, mainly of Bacillus and Clostridium families, although there's at least one other family that makes endospores. But mainly these 2 families make endospores. And what happens when the environment starts to become detrimental, the parent cell will begin making an endospore, and the endospore will be made inside the parent cell right here. So there's the endospore, and there's the parent cell. And then when the environment becomes so detrimental that the parent cells die off, the endospore can survive. It's not reproduction because one parent cell makes the endospore and then dies, and we only get one endospore. So 1 into 1 is not reproduction. The endospores are highly resistant to most substances, like they're resistant to desiccation, heat, chemicals, radiation, and they can survive conditions where the vegetative cells or the parent cell cannot. We call endospore formation sporulation or sporogenesis. And then when the endospore returns to the vegetative cell, we call that germination. Germination of the endospore to make the vegetative cell. The vegetative cell is the normal growing, reproducing, metabolizing cell. The endospore does not grow, does not reproduce, and has very low levels of metabolism compared to the vegetative parent cell. Any questions about any of that? The main point is endospores are not a reproductive strategy. They are a survival strategy. Endospore formation happens a little differently than most bacterial reproduction. First of all, endospore is not reproduction. I've already mentioned that. But the cell, when it's going to form a endospore, actually, oops, let me go back, get this out of the way. The cell actually goes through division, but the cell does not divide equally. There will be a smaller division which will go on and form the endospore, and then the rest will be the remains of the vegetative parent cell. So this part here will continue being the vegetative parent cell. The DNA does get replicated, and one copy moves to the other side of the cell to be the copy of DNA in the endospore. The plasma membrane will then divide or form in between the endospore and the parent cell. We'll get 2 plasma membrane layers. You'll put peptidoglycan layer between the 2 membranes. You will then form a spore coat on the growing endospore. And then this parent cell will die, and that will release the endospore. Any questions about any of that?
I had a question about the inclusion bodies. Yeah, for electron microscopy, isn't inclusion also a word used for like things that are hard to discern or something like that, or I'm confusing it with another word.
I think you're confusing it with another word. An inclusion is something inside the cell that the cell is accumulating, and it accumulates at such high levels that it precipitates out of the cytoplasm, and then you can see it separately in the cell as an inclusion. Okay.
Okay. Oh, maybe I'm thinking of a artifact. I think that's what I.
All right. Yeah. An artifact is something we're not sure what it is.
Okay, I see.
Okay.
All right.
All right.
Thank you.
If there's no further questions, I'm going to end it here and I will I talk all about endospore formation. Yes, I did. All right. So let's end it here and I'll see you in the lab at the regular time. Okay.